państwa przyznanych przez ministra nauki i szkolnictwa wyższego. Dofinansowano ze środków ministra kultury i dziedzictwa narodowego pochodzących z Funduszu Promocji Kultury, Państwowego Funduszu Celowego. Partnerem wydarzenia jest miasto Kraków. Copernicus Festival został objęty honorowym patronatem ministra nauki i szkolnictwa wyższego oraz prezydenta miasta Krakowa Aleksandra Miszalskiego. And now we can safely switch to English, and I have the great honor to welcome and introduce on stage Professor Zuzana Hofmanová with the lecture Identity, Ethnicity and Genetics, Who Are We? And after the lecture, there will be opportunity to listen to a conversation about these subjects and also ask questions. So if while listening you're thinking of good questions, you will have the chance to ask them. Now please welcome on stage Professor Zuzana Hofmanová.
So, hello, good afternoon, good evening. Thank you very much for coming. I would like to very much thank the organizers for inviting me to this beautiful event and to this beautiful city. I've been having a really great time here, and I hope you will enjoy my talk at least a little bit as well. So I will be speaking about identity, ethnicity, and genetics. And for some of you, this might be something that's a little bit strange, like genetics, why ethnicity, how it's connected. So with identity, it might be a little bit easier. I mean, it's kind of easy to imagine that you can identify an individual by their genetic information because, you know, you can imagine some kind of crime TV show where they leave something behind and researchers are then identifying who that is. With ethnicity, it's more complicated, and it's more complicated for very, very good reasons. So ethnicity also is often defined as having shared culture, and that is often related to common ancestry and shared history. But this relation with this common ancestry is not so clear. And it must be said that kind of this, let's say, unclear connection between ethnicity and ancestry and genetics is a little bit explored by certain companies that are trying to kind of sell you information about your ancestry, but actually are trying to give you information about your ethnicity or the other way around. So my kind of favorite example in that is that some of these companies will like to tell you that you are 15% Viking. And that's, you know, relatively funny because Viking is actually a job occupation. So it's like saying that you are 15% social worker or 15% doctor. So that's kind of the level of information you might be getting. And I would like to kind of explore this connection between ethnicity and genetics and ancestry on not on kind of modern DNA, but on something else, on cemeteries, cemeteries that are found during archaeological excavations. So this behind me is an example of one of those. They are often found when something is being built. In this case, it's a large-scale excavation that has been opened because of a building of a highway, but it could be also like during a building of a building. And the ground will be destroyed. And there is information in it. And that's information that is worth preserving, and that's why there is archaeological research. And I would like to kind of tell you a little bit what kind of we can learn from this information that is being preserved before it's destroyed by the building itself. So here just an example of the painstaking work the archaeologists are taking while excavating. It's a hard job, and but it brings great finds. And so one of the things that you might think of, like what does such a cemetery could tell us, is that sometimes like there would be graves one like above the other, and from this you can say who died first and who died later. Or some graves could be very close to each other, and you can think, okay, maybe these people were in some way connected in life, maybe they were related. So then you can kind of think about the things that were buried with that individual. And there is much to be learned from these artifacts. You can think where they were made, what kind of material they were made from, how they were made, how they were used. But you can also think about certain artifacts or certain objects, like in this example there behind me, it's strap ends from belts. And if some of these are very much the same because they come from the same forms, and you might think, okay, so these people that have the same object must be connected in some way, maybe related, maybe, you know, having some kind of other connection. Then you can think about the actual skeletal remains. So there was a really great workshop that maybe some of you have been participating about what the bones can tell us. So it's possible to estimate height, robusticity, sex from the skeletal remains. It's possible to kind of discuss the diseases and injuries that those individuals had. And in some cases, there are certain markers that are like hinting at, like, you know, that these people could be related. And you can kind of understand this because, you know, in your own families, maybe this shape of ear is, you know, more common, or this shape of eyes. And that's something that, you know, you understand. On the other hand, you also understand that, like, not everybody in your family has the same ears, and maybe actually somebody completely unrelated to you might have very much the same ears. I don't know why I picked ears, but here we are. And this tells us that kind of this similarity on these markers is like useful, but can't be used on this kind of individual level of you are not very like sure about this. And here I would like to kind of make a little disclaimer that people have been trying to find similarities on these like kind of skeletal markers in the past, and also on the grave goods in the past, in terms of like similarities between different populations. And much has been done in like beginning of last century in terms of like trying to identify the Aryans, etc., etc. There's quite some bad history in it. But I hope I was trying to explain that it doesn't work on this individual basis. So it's not only dangerous, but it actually does not work. So what else we can kind of learn from the bones? And one thing that can be done is the isotopic analysis of the bones. And when we eat, like kind of the material of the animals or the plants that we consume is getting to us, and with it the isotopes of those individuals. And from that we can understand if like somebody ate more protein or less protein or different types of plants. And this kind of, again, could tell us, like, if two people had similar diet, maybe they were connected in some way or form. Or if somebody had ate much more protein, maybe that individual had a higher status in the society because it was difficult to get proteins, it was difficult to get meat. Or a different way how you can think about isotopes is isotopes that usually, or it's assumed to get from water, that is connected to the geological background of the area. And if in those bones that you are studying, this isotope ratio is different, then possibly those individuals were drinking different water, hence they're probably from somewhere else. So it can tell you something about like where those individuals were from, local or non-local. But I'm a geneticist, and I will be mainly telling you about another kind of molecule that you can study in the bones, and that's ancient DNA. And you might have heard before about ancient DNA. It's both from humans or sediments or animals. Like ancient DNA is defined as DNA that's at least 150 years old. And there's a lot of information we can learn from that. We can learn from it, like there's ancient pathogen DNA. So if in that DNA that we are working with, there is a pathogen, we can tell how those people died and how our immunity evolved. And we can even say that if two pathogens were the same, maybe again those people kind of got it from each other, so they were somehow connected in the past because, you know, they must have get it from the same in the same way. But the main focus of my talk will be ancient human DNA. And I will be speaking about genetic similarity, how we are kind of similar, you know, in some regions, to some regions more than to some others. But I will be also speaking a lot about relatedness, how we are really closely related to each other and how that can be investigated from genetics. And another way how you could know ancient DNA is that in 2022, Svante Pábo, a director in our institute, obtained a Nobel Prize. We did properly celebrate by throwing him into a pond, which is usually done for PhD students when they pass their degree. And from now on, it's done whenever you get a Nobel Prize. So, and, but, you know, on the way to his degree, or to his Nobel Prize, there was a lot of hardships he had to overcome. It was not easy because our field went through this kind of hype cycle. So this hype cycle is something that's quite common for many technologies. There is something new, everybody is extremely excited about it, they think they can use it for everything, and then slowly they realize that there are limitations, and then people are very disappointed. And then slowly you kind of get back to kind of this plateau of productivity. And I would argue that we are, at the moment, with ancient DNA in this plateau of productivity. And the reason why there were these like kind of inflated expectations where the problem lied was that all these like older ancient DNA studies actually proved to be not of that animal or human that was analyzed, but actually like modern human. Often the researchers themselves, not necessarily Svante himself. Because in our samples, there's actually a combination of things. So we analyze modern DNA or an ancient DNA and also the soil DNA. So if you get DNA from the bone, like there was also water running through that soil, and you would be surprised how much of human DNA is everywhere. We are so many, and there is so much of us and of our waste. If you know, you would open here little like a falcon and wait it like open for a bit, and close it after a while, you could actually get DNA molecules from it, because that's how much DNA is in the air, in the water. So that's why when we are working, we have to work in these very protective environments, in laboratories, so we ourselves do not contaminate our own samples, because there is more DNA on our skin than there is in our samples. And we also pick our skeletal elements, the skeletal parts, wisely, or we try to. So we often analyze petrous bones. So these are parts of temporal bones, and this is where your inner ear is, and this is the hardest part of the body, and that's why it's the most protected from kind of the environment that would degrade it, but also from the modern human DNA that's around it. So then, okay, so we drill into it, and I didn't say there is actually this little hole, so that's artificial. That's the one that we have done during the sampling, where we go inside it and take sample of 25 milligrams, like extremely small amount of bone powder, that then gets dissolved and cleaned until only DNA stays in it. And even despite all of this, there is still some modern DNA. It's just inevitable. So we have to be extremely careful with our samples. We have to check that there is actually only one individual in it, because if there is more, there is a problem. And we also have to check if there is a damage, because, I mean, bad for us, the DNA that we study is damaged, is very short, but good for us is that this means that it's distinguishable from this modern DNA that's around. So this is kind of like a happiness and sadness and happiness. So, but I mentioned that these DNA is degraded, but that also means there is very, very little of it. And it's actually quite hard and very expensive to study. So what we can do, we need to somehow kind of increase the fraction of the human molecules so that we can kind of get to them a little bit more. And so we use something that we often colloquially call capture, which allows to kind of with magnetic beads to bind to the probes with human specific regions, human specific DNA, and that way we can enlarge this fraction and study it better. And this is great, not only because it's cheaper and we actually can study some samples that otherwise would be completely unstudyable, but also because the amount of data that we are handling is much less, because it's a lot of data, especially if you consider that we have a lot of samples. And most of our analysis now actually does not happen in the lab, but we spend most of the time behind the computer like everybody else, I guess, kind of on computation, which is usually happening on computer clusters and some basement of some institution. And just for you to a little bit understand how much the field has changed in very few years. So here I have an example what was good enough for publication just few years back, or okay, not so few, but some years back. And so it was possible to get to a science publication with just 3 genomes, while just, approximately 5-10 years later, you had to have kind of 40, etc. It was increasing really fast. And now we have a project where we are analyzing 6,000 samples. Yeah, so just kind of in span of very, very short amount of time, kind of this has blown out. And the main reason for this is not that, we did improve our methods, but it's mainly thanks to the improvements of the sequencing technologies that are also improving medicine and many other fields. So this project that I will be speaking about is kind of brainchild of Johannes Krause, Walter Paul, Patrick Geary and Tivadar Vida. And it's a large project that is trying to study the period from 400 to 900 of Common Era. And I'm coordinating the genetic analysis in this project, and here I just would like to thank you to my team that is helping me, because it's a lot of work and I would definitely forgot to thank them in the end, so I'm thanking them now because I'm very grateful for their contributions. So what we are actually doing here. So here is the list of sites. So these are the sites, these are the archaeological locations that we are studying. These are the cemeteries. And to this, we have added few more points because we have very soon after the project began, or actually even before it, realized that you really need to have also study like surrounding area to understand Central Europe as a whole. So what is our strategy? Why did somebody, European Council, have decided to give us kind of quite some amount of money to do this? What is kind of different about that? And the difference is that we are working very closely with historians and archaeologists on the project from the beginning till the end. And also that we are working on complete cemeteries. So before, what was usually done was that you study a few samples from one period, few samples from other period, and then if you see a change, you see a change. But we would like to kind of understand this process more. We would like to see what's happening and study this systematically. So that's one innovation. And the second innovation comes with the technologies I was thinking about. That we are really trying to get to every sample, like approach, leave no sample behind. We are not at no sample behind, but we are really getting there. So with kind of really high success rates for what is kind of usual for ancient DNA. So we are studying early medieval period. So why is that interesting? So if you look at the first millennium in general, and you will look at the beginning of it and the map of the Roman Empire, you would see some borders. And then if you would go like 1 century later or 1 millennium later, you would see much difference. So there's been a lot that has been happening in that millennium, and a lot of it is actually quite crucial for our understanding of also events afterwards. But you might think, wait, like historians and like first millennium, like there's a lot of like historical sources, a lot of books, and I learned about it at school. Like we know what has happened, no? And that's kind of a catch. Like it's like knowing and really knowing is a different thing, and the written records that we have are actually very limited. Like they often represent a viewpoint or particular historian from a particular city or for particular realm, and they don't cover all the people. So often these would be people from the church, so we know a lot about development in church, but we, for example, don't know much about traders. So there is a very like kind of what section of the society is covered. For some centuries, we have very few mentions of women, for example, etc., etc. The archaeological record exists and is wonderful, but it's very hard to connect it to these historical records so easily, because there's often like issues with dating, etc. So one example where I would like to kind of tell you like what can be difficult are Avars. So Avars is a group of people that appear in historical sources in 6th century. So they are writing to the Byzantine Empire that here we are, give us money so we don't attack you, in kind of a smaller or how to say, easy-fied version of what they have written. And but they were actually extremely important because they have set up this Avar Khaganate that lasted centuries in Central Europe. And but we actually don't know much about them because they didn't leave any of their own historical records. So we only know about them like from their enemies, and they were not exactly writing, you know, nice things, because you don't write nice things about your enemies. But we have a huge archaeological record. There's more than 100,000 graves that is kind of attributed to this period in the Carpathian Basin. And one of the things that we don't know is that we don't even know where they were kind of coming from. How did they appear in Europe? So where there is the cross, this is where they are first mentioned in the historical records. But they themselves claim that they come from East Asia, that they are kind of the descendants of the Ruran Empire, which was a very important empire in Asia. But the contemporary authors, the Byzantines, the Byzantine historians are writing that they are not so sure about it because the Persians are telling them this is not true, that the Avars are making this up, and that actually they are coming from elsewhere. And this kind of idea, this kind of a, let's say, mystery has been discussed in history since then, so for the 1,500 years. So what have we done? So we have looked in the published literature for ancient DNA data and collected ancient DNA data from the pre-Avar period. And then put it on a PCA, principal component analysis. And on that analysis, which is then the one on the bottom, there is like each dot is an individual. And if those dots are closer together, these individuals are more genetically similar. If they are further, they are less genetically similar. And on the map, you see like in blue the Carpathian Basin, in green the steppe, and in yellow or orange the East Asia. And the geography is very much similar as the genetic similarity. And that makes sense. This is quite common for almost any species. And the reason for that is that you are just more likely to reproduce with somebody who lives close by than with somebody who is very far away. So not that unusual so far. So then we went to the Carpathian Basin and we collected skeletal remains from graves, from very elite graves, with the idea that the elites will likely be the ones who migrated, if there actually somebody or if anybody migrated. And then we compare them with this published data. And what we have kind of seen is that the red and orange, our new samples, are kind of plotting on the same spot as those East Asian samples, even though that they were found in Carpathian Basin. So that tells us that indeed this individual from these Avar elite, Avar period elite graves were actually having this connection to East Asia. So kind of they were right. It was not the Persians who were right. And I mean, it's true that the situation is a little bit more complex. There's also genetic influx from the area that the Persians did mention. Okay, so Avars. But you might be kind of thinking, okay, so we collected these kind of new sites, new regions with the help of quite a few researchers, and it was not easy because in many of these areas cremations were actually more common in this period than inhumations. But why we did it? And you know, if you look at these areas, they have something in common. They come from Slavic-speaking countries. And you might kind of wonder why we did that, why we decided to look into these kind of like Slavic associated burials. And what might surprise you is that there is actually quite a lot of debate about origin of Slavs. And that, because maybe it doesn't, but it surprised me when I entered university that things are not as sure as given, and that what I learned in my textbooks might be different from somebody else learned in their textbooks in different country. And this is because of these limited historical records I was mentioning. We know very little for sure, and there are very contradicting interpretation of these historical records. And there's also large space, so there's also geographical variability as well. And what's kind of worse to some degree is that this prevents us to studying these populations more, because like always we are kind of going back to this question and not kind of studying other things that are also very interesting in those regions and areas. So you might wonder, like, why is it a debate? So and there are several reasons, and kind of you may think about from the point of linguistics, where Slavic languages are actually very similar, and it's been quite discussed how exactly that tree should look like. You might think about from the point of genetics, where all Slavic-speaking modern populations, and that's the graph below, have been found to be quite similar to each other. And that has been interpreted as A, because they have a common origin, or B, because the population sizes in the regions were small. And actually B is correct, but we don't know if A is correct as well, but B is correct. There were lower population sizes. And then comes archaeology, which is showing this kind of a archaeological phenomenon that's appearing in those regions that we are discussing later on, Slavic-speaking regions. And but it's kind of unclear where this phenomenon originates. And there are several theories. And often, because there's a lot of work from Polish researchers, often like this debate is framed from the point of view of autochthonous origin, meaning from Poland, allochthonous origin, not from Poland. And but there's also theories about that the kind of a Slavicization happened mainly in the Danube border, and then the language has spread only as a language, not necessarily as the people. You can think about Hungarians as an example how that can be done. And just a little kind of, to make it even maybe more complicated, is that I was quite surprised at the reactions we got when we discovered this quite small piece of a cattle rib on which there were runic inscriptions. And these runic inscriptions, which are connected to Germanic language, have been found in a context of this Slavic-related culture, of so-called Prague-type culture. So so this already kind of, for some people, this kind of questioned if really, or if they thought that we are claiming that this culture was actually German-speaking. That's not what we were claiming. We were just, reporting a find, and you might imagine that I'm speaking here in English and I'm not English, so you can have things related to certain language at some other area, and it's fine. Like it's fine. So So what have we done? And I will be presenting here research that's not published yet, but has been accepted. And it must be said that it's a work of many, many people, led here by Joschka Gretzinger. And it also must be said that there are researchers from Warsaw, from Krakow, and Rzeszów, and actually many other places in Poland that are involved in this research. So what have we done? So again, we collected the published ancient DNA data and also added quite a few new to it. So this is the colorful dots. And you might kind of notice that there is a gap in our sampling, and that's not because we would wanted it, it's because those are the cremation period. So it's kind of hard to get samples when, you know, they burn them all. And I mentioned that these samples have been published before, that's true, but interestingly, some of these studies have actually come up with very contradicting interpretation of this data, which stems from the archaeological context or lack of thereof, or because of actually not having enough resolution or enough samples. So our next innovation was trying to kind of improve the resolution. And to do that, we have collected data from like modern day people that live today, and that you can see behind me again on this kind of a principal component analysis. And because there were so many, it was possible to kind of differentiate different regions and kind of little bit understand the variability of Europe today, but possibly also in the past. So on that, we have put the genetic samples that we have obtained from this period before the presumed kind of influx of Slavs, so pre-600 of Common Era. And this placement makes sense. So the Croatian samples are kind of close to Italian samples. I mean, the area was under Roman Empire, and there is a lot of Iron Age kind of genetic variability there as well, the same one. Russia, that's also like, there are probably Uralic speakers, that's also quite clear. For Poland, it's quite interesting because these samples are not where modern day Polish people are placed on such a space, but actually kind of on Scandinavia. This is connected to Wilbar culture that has actually links to the north. For Eastern Germany, it was also quite interesting because there was a lot of this similar ancestry as in Poland, but also quite some individuals had the southern ancestry, which possibly meant some kind of a connection to the Roman-speaking areas, or sorry, to the Romans. So I kind of clicked too fast, but here you see what has happened after. And basically this quite large genetic diversity have collapsed. Not completely, as you see, the Croatian samples are still shifted a little bit more to the south. But overall, in all these regions, people became quite similar. So from that, we conclude that demographic shift has played a role, quite significant one, in all these regions when it comes to kind of the change. And since from historical records, there was appearance of Slavs, and there is this archaeological horizon, we can start to connect these events. And we didn't only compare to the modern samples, we also compared to the ancient samples. The closest samples were actually from Iron Age Latvia and Bronze Age Latvia, so these were kind of the regions, though it must be said that we are missing a lot of samples from Eastern Europe, especially Belarus. It's hard to sample for, I think, understandable reasons. So kind of to put this evidence from the ancient and from modern and from various types of analysis together, what we came up with is this kind of model, which means kind of a understanding at this moment of time. That is that there was this Baltic-related ancestry that at some point, and maybe over some longer period of time, have admixed with southern ancestry and created this gene pool that has later on spread to the rest of the Europe and influenced it very heavily. And you might kind of see that I try to speak about ancestry, not about people moving. And because one thing that should be said is that it's not like that that the local population had no influence. You already saw it on this Croatian example, but we see it on all the other examples too. There is always some level of continuity as well. There is always some level of admixture as well. And but what is quite interesting is when we compare different modern populations. So, for example, here Sorbians with modern Czechs, where the Sorbians actually keep much more of that kind of ancestry than Czech speakers. Sorbians have it more, so they're kind of let's say more conserved in that regard. And just a little note, not only the ancestry changed, but also social structure has changed. People before were not so related to each other at the cemeteries. They were much more related afterwards. So, is it simple? Is it so, Avars, Slavs, is it easy? And answer, no. And I would like to tell you a story of mainly two sites. They are both very close to Vienna in Vienna Basin. One is called Medling, that's actually in Vienna itself today, and the other one is Lebersdorf. And we sampled quite large amount of samples from these cemeteries, like all these cemeteries, complete cemeteries. And what's interesting about them is that they are just 20 kilometers to each other. They are very, very close by. And archaeologically, they are very similar. So they are both attributed to the Avars or to the Avar period based on the strap ends and all the other things that those people were having in their graves. And they are also supposed to be dated to the same period. But when we did the genetic analysis, and here I would like to remind you of that genetic analysis I showed you about Avars. So if you are more to the right, you are much more East Asian. When you are to the left, you are much more European-like. We are very, very surprised because they are so different, as different as somebody from Europe and somebody from Asia today, genetically. But they were just 20 kilometers from each other. So we're thinking, okay, so maybe there is some issue with the dating. Maybe it's like a process where like slowly the East Asian people are admixing with the modern populations until this disappeared. But actually there were close relatives between those sites. So we could actually say that this was approximately at the same time. It couldn't have been kind of completely far apart. And just a note, when we speak about the Medling side, about the European ancestry, if we zoom into that ancestry, this ancestry actually has this northeastern European component. This component that, you know, we were kind of before showing for the Slavs, for the Slavic related cultures and burials. So what's happening here? So, I mean, Slavs and Avars had connection. We know that in historical records. So maybe there was some kind of hierarchy between them. So maybe there was a social status difference between one side and the other. But we couldn't find any. Like there was not on the grave goods, not on the diet. They were looking extremely similar. So we are thinking maybe the Lebersdorf individuals, the ones with a lot of East Asian ancestry, maybe they were like soldiers that were supposed to keep the region in check or something like that. But no, there were no weapons, no injuries that would relate to that. So we looked at other types of analysis. And here I will be speaking about something called IBD, identity by descent segments. And that is something that if two people share between each other, that indicates direct relatedness. Because it's a very long stretch of the same DNA that's shared, and it just can't be broken or wasn't broken out in recombination. So by the length of that segment, we can also say how far or close these people are related. And one way to represent this huge amount of relatedness are these relatedness networks. So each here, a dot is an individual, and if they have a connection, they have this direct relatedness. So a line, connection, dot, and individual. And what you can see that our Lebersdorf, the East Asian with the East Asian ancestry, is quite different from Medling with the European-like ancestry. And Labrador is actually much closer to other sites, to the sites that I was a little bit mentioning before when I spoke about the elite sites in the center of the Khaganate between Danube and Tisa. Another way how to represent this is this kind of a connection to these regions. And I just wanted to show you the network so you see that there is a lot of information that goes into that picture. But another thing you might have noticed in this picture is this kind of like these clouds of the same colors, this like lot of relatedness within the sites. And that's very interesting. And this relatedness was very close, many first degree, many second degree related. So that means that we could reconstruct pedigrees. And I couldn't tell you how much enjoyable, or I couldn't tell you enough how much enjoyable this process is of putting things together and of kind of getting to, I can understand this. And when like kind of these large pedigrees start popping up, we were like beyond excited. And because this can tell us a lot about the people who was kind of connected to whom. And one thing that's kind of should be noted is that only 6 individuals from all those individuals were completely, fully unrelated. All the others were actually either in a pedigree or connected to them. So that was quite unusual. One thing you might think about, did it matter to them or did they know and did they care? And one way to kind of look into that is look at the burial positions of the various pedigrees and try to look if the individuals that are related, if they were buried together or not. And there comes really interesting stories from the burial positions as well. So one example is this, and this is from slightly different cemetery. That's one of those from Danube-Tisa interfluve. So here is an example of a like subsection of that pedigree. And the way you read that pedigree is that the square is a man and the circles are women. So this man in the middle, and I hope you understand me even without the pointer, and is having had 2 partners in life. With one, he had 2 sons, and with the other, only 1. So, and then he is buried in the middle, surrounded by his sons, and also one of his partners, while the other partner is not buried there. And you might already start thinking, why, and etc. But one thing to note is, like, nobody is burying themselves. It's usually the community or those alive that are burying you. So maybe there lies our explanation. But what we saw is, and I spoke about the man and his sons, and this is kind of a thing that we see again and again. So all these pedigrees are connected father to son to son to son, almost all of them. So they are almost strictly patrilineal. On this pedigree, we have only one, that's the little pink dot in the middle, one woman that's at the same time mother and at the same time daughter. All the others, all the other daughters are at certain age disappearing from the site. They're probably getting married to other communities. So, despite all of this and all this relatedness, there is no consanguinity, no inbreeding. But we do find interesting things. So we do find that these levirate unions where brothers have the same partner, where father and son had a child with the same woman. It was not the mother of the son, but just another wife. And these kind of levirate unions that we see in the pedigrees have inspired historical research that confirmed that this was indeed happening in steppe societies. And just one little note about the women, the daughters that are disappearing. It's so interesting that we can see the age when this is happening. Because and it's not the age that some people expect in early medieval period, they get married or very, very young. That's not what's happening. They're actually married like around 18, some of them 20, 21. This is when they are becoming like the first mothers, but also when they are not anymore buried at the cemetery where they are coming from. And just a little note, there's also quite some studies about that are going to come about pathogens of these people. There we can see kind of how pathogens are actually clusters within the families and are probably spread within them. in this case, hepatitis B virus, usually thought as sexually transmitted disease, but in this case, we think it might be not likely, or might be quite likely, that they are getting it during the birth from the mother, because that's actually quite common transmission in kind of pre-industrial societies. So we have our Labersdorf, we have this kind of understanding of the social structure of this community. So was Medling different? Was the other side different? Because they had different ancestry. So does that kind of, they were already living completely differently? And the answer to that is not at all. Not at all. Actually, the other cemetery, it's so hard to show that pedigree because it's 350 individuals connected in it. So it looks like a little ants on the presentation slide. And what we then see is, because we can then connect them, we see 6 generations of this reproductive barrier between those two very kind of geographically close related, culturally the same sites that also have this same pattern. So also the patrilinearity is extremely strong here with levirate unions, as see here, where 2 brothers are connected to the same woman with whom they have sons. And interestingly, those sons that are half-brothers, but actually more half-brothers because they have their fathers were brothers, have also children with the same woman. The family dinners might have been interesting. And I'm kind of laughing about it, but for such communities, it has been extremely important social practice that kept the women inside the community, protected the children, and also served as kind of a continuity of the leadership. But about the women, you can see that they all are coming from elsewhere. The female exogamy was very strong. And I'm not saying that there is no matrilinearity. There is actually quite some, but much less of it than in terms of patrilinearity. So we didn't get very far. So we got to the point where also the social organization is same between these sites. So what is then different? What makes them different? And what keeps them different for so long? And answer comes from these women that are kind of coming to these communities. Because you remember, I told you about these kind of long distance relatedness connections to other regions. And these women were coming from different regions. So the Medling individuals were kind of, the community has been getting the women to the community from communities that were kind of having similar ancestry, whereas Labersdorf was more connecting to the communities that were living in the center of the Khaganate. And that means that these different marriage networks, and you must remember, marriage at the time was economic, political proposition. So it was these basically political connections were creating the ancestry difference. So it's the culture that actually influenced the genetics, not the other way around. And that kept for more than 250 years in kind of heart of Europe, close to Vienna, these kind of communities that were of East Asian ancestry, kind of still extremely East Asian-like. So what we can kind of conclude from all of this. So one conclusion that we have is that there is or at least we can see this kind of demographic shift that's related to the Slavic associated archaeological culture, but also to the later on Slavic speaking regions. And we can see it. And I was not speaking about it too much because it's still a paper to be published, but it has a continental wide impact that's even to the regions which are no longer Slavic speaking quite consistent. No matter that some people might not like that. And another conclusion is that, okay, but the ancestry is only one component, only one thing in a way. So when combined, combined with other lines of evidence, we get kind of more complete picture. We get understand that affinities, ethnicities, identities, they're changeable and they're changed for sure. and also that influence was kind of constructed by people, by behavior, by reproductive behavior of people. And here, kind of when you think then about who is an Avar, and sometimes I get asked, so the meddling people are like not real Avars. It's the Labersdorf people that are the real Avars, no? And I'm like, for 6 generations, they've been marrying and living in particular way. They've been burying in particular way. They've been, in some cases, not in this, but in other cases, living probably in the same settlements, maybe even in the same houses as their ancestors. So are we really kind of like, is it okay for us to say that because their ancestry was different, ancestry of which they knew nothing about, that they actually were not real Avars? I don't think so, but you make your own conclusions. And kind of another kind of thing connected to that is I spoke about this European-like ancestry, that it's rare, but it does happen that there are these women that are coming also from other sides. And so that means that there are some like patrilines where at the beginning of it, we have somebody with a lot of East Asian-like ancestry, but 5 generations later, that ancestry is only 5%. And that's, I think, if those 2 people live today and send their DNA to these companies, they would be told that they are somebody else, that they are not connected to each other. And again, we are speaking about people that lived for generations and generations at the same time, in the same way, being dressed almost the same way as their ancestors. And so that kind of tells you something about like how that ancestry is important, but it's not a destiny. It's not a thing that you can rely on to kind of define your ethnicity. And I hope to show you at least a little bit how kind of the important thing is to connect to other disciplines. So how we are connecting to history, where in historical records we are finding clues about the social practices we observe. Also that these kind of pedigree-like studies are extremely important when we want to understand a spread of diseases in the past, and also immunity, because how immunity was passed and how people were kind of in contact with pathogens over time. Not to mention that the ability to tell the generations apart is extremely important for archaeologists, for dating, and for anthropologists also to kind of understand the size of the communities. But there is like one more thing that I didn't mention too much, and that is that we indeed learn from Engineering Day about and from archaeology together about communities, about populations, about social practices, but with this example I was showing you before, we also learn about life histories of individual people, and it's not, sometimes they are not like easy histories, not simple histories. We find a woman that is buried close to her 7 young children that very likely died before her, and that's not like, it's like you can always kind of think about, did people knew if somebody was related to somebody else? But there are some relationships that are understandable and we can connect through them from today to the past. And these people have survived this and had other children and lived through this preserved. And often there were books and books written about kings and aristocrats, but actually most of our ancestors were not bad. They were kind of more of these normal people that don't usually appear in historical books. And this is one way how we can kind of look at them and think about them and maybe in some way honor them. And as I said, like this can be done with other disciplines and with kind of understanding of humanities and and genetics and statistics together, and I kind of like that's something that was mentioned in the introductory talk of this festival, of this kind of how kind of connecting various ways of knowledge is important, and that can tell us something about mysteries, in my case, mysteries of our past. So with that, I would like to thank you very much for your attention, and I would like to thank the organizers and everybody who helped with that research. Thank you very much.
Thank you. Thank you very much. And I think that we will wait with the question bit for the meeting on stage. So in 15 minutes, we will meet again. We'll also introduce Professor Małgorzata Kot, who will join us on stage, and we will have a discussion. So if anyone has questions, I think this will be the proper moment to ask them. So thank you very much again, and don't leave. You can walk around, enjoy some fresh air, capture the festival catalog, which is to the left when you leave, and then come back to talk. Thank you.